of conference organized to celebrate the 75th anniversary of our birthday of our institute. And Dr. Monica is a professor and chair of the Neurobiology and Anatomy Department and adjunct professor in ophthalmology and visual sciences at the University of Utah. After attending McGill University, she attained her PhD from the University of California in San Francisco, where she did also a postdoc. And she received many awards and prizes, and she one of them is the fact that she's a founding member of the first Catalyst for a Cure Research Consortium and currently serves on the advisory board for the Catalyst for a Cure Biomarker Initiative. She is an established neuroscientist, and she has published many influential scientific papers in developmental biology, mainly on the regulation of neurogenesis by transcription factors and extrinsic signaling pathways, and also on the epigenetic regulation of progenitor proliferation and differentiation. Well, in summary, her laboratory is focused on understanding the molecular pathways controlling neurodevelopment, but also degeneration in the vertebrate retina. And her recent emphasis is on microglial functions in both fields. So this is the subject of her talk today. So thank you again, Monica, and I hope everything works well for now.
So Mariana, I'm going to ask you just to moderate the questions because
no, feel free to make your presentation. We will take notes if the questions show up during the presentation, and after that we can open for questions.
Okay, and I'll just tell you when to advance. So thank you.
I'll turn my camera off now so it doesn't disturb this, and let's see if will work well. Okay, so whenever you want to start.
I don't see the slides yet, so you're going to have to share.
Yeah, let me try again because it is just weird. But let's just open it. Just a moment.
We've learned a whole new set of skills in it.
Oh my God.
I haven't mastered the Google platform, so I apologize.
Let me see if it works now. So, is it there?
Nope. I see all the faces.
It's not.
There we go. Now
it's going to open up.
Yeah.
Is it?
Now I see it. Yes. Thank you.
Okay, perfect.
So, so I've had an opportunity to thank you all. And again, this is a tremendous honor. And I really am pleased to be able to join you in this celebration of your institute. So Mariana, will you just advance the slide and the text should, so that I show this image because this is an image from my friend and colleague, Dr. Alejandra Bosco. And it shows the retinal axons in cyan migrating towards the optic nerve head, and then the microglia in red. And this is a flat mount view of a whole mount retina. Alejandra is an Argentinian scientist who trained at your institute from 1992 to 2001 with Dr. Rafael Linden. She has spoken often about this training as a transformative experience in her career. I'm going to be discussing her work later in the talk. She's been a member of my team since 2006, and she's now Research Associate Professor in the Department of Neurobiology and Anatomy here at the University of Utah. It's clear her training was impressive. She's just brought tremendous knowledge and skill and passion for science, and it's been a real privilege for me to work with her. And so I feel a connection to you all there. And I just wanted to highlight some of her work, and I'll be talking about that more. Next slide, please. So as Mariana mentioned, I'm going to be talking about the retina, and I know that this is a diverse audience. Many of you are deeply knowledgeable about the biology of the retina. But it's a wonderful model system for asking a wide range of questions related to both development, adult neural function, as well as degeneration. And we know that during development, retinal neurons are generated, and they have to be generated in appropriate numbers in order to establish connectivity and function appropriately. These cells need to be maintained throughout aging. And then we know that specific populations of neurons are vulnerable in disease. So, for example, retinal ganglion cells, which are the major projection neurons from the retina to the brain, are particularly vulnerable to degeneration in glaucoma. And so this image is just a beautiful image from my colleague Rachel Wong at University of Washington, just showing the diversity of cell types. Next slide. And so much of my lab has been focused on trying to understand how retinal progenitors differentiate to generate these diverse cell types. And so on the left, you see a scanning EM image from Kathleen Sulik showing early stages of the developing retina with these retinal progenitors that will undergo many rounds of cell division. And then in the center, you'll see that these cells begin to exit the cell cycle and differentiate in mouse during the embryonic period, starting at about embryonic day 10.5. And there are successive overlapping waves of cell genesis. So retinal ganglion cells are always the first cell type to be generated. And then all the other cell types of the retina are generated in a very defined sequence that's highly conserved, with Microglia among the last cell types to be generated. And this results in the beautiful differentiated cell types that you see in this image from Don Sakaguchi on the right. And so we've learned a lot from work of many labs about the gene regulatory networks and transcription factors that regulate the genesis of these cell types. And we know a lot about the connectivity and function, although there's still much important work that is ongoing. Next slide, Mariana, please. But one population that was not originally thought about in the context of development are the microglia. And this is an image from Ali Bosco. This is a flat mount view of a mouse retina, an adult mouse retina. And this is from a reporter mouse, fractalkine GFP reporter mouse, where the microglia are selectively labeled with GFP. And so you can see the incredible abundance and organization of the microglia within the retina, and they occupy three major layers within the retina. So they are really a key component of adult retinal function. And so we need to really be thinking about how these cells participate in both the development and function of the retina. Next slide, please. And microglia, unlike all the other cell types of the retina, are actually populate from outside of the nervous system. So this is microglia are a resident innate immune cell population, sort of resident macrophage-like cells within the nervous system. And early in development, they originate from yolk sac precursors that during a process of primitive hematopoiesis that then colonize the central nervous system, including the retina. And so this is just a whole mount image from Sarah Anderson in my lab showing already by E12.5 the microglia are present and have populated the retina. So we're very interested. They're clearly present at the time that all these different cell types are being generated. And so we are interested in understanding how they actually might contribute. Can you advance? And so work in the brain has shown that microglia can play diverse roles in CNS development. So they're important in their surveillance function. They're detecting perturbations to the microenvironment within the nervous system. But there's evidence that they can regulate programmed cell death. They're important for phagocytosis. They regulate neuronal plasticity, synaptic pruning. And we're beginning to learn some of the molecular pathways that are involved in that. Advance. But the microglia function during development of the retina is really still not well understood. And I would argue that the retina is an ideal system to really address this, because many of the steps involved in both the genesis and connectivity of those cell types are fairly well defined in this system. Next. So the first part of my talk, I'm going to ask, how do microglia shape the developing retina? And this first work was spearheaded by Sarah Anderson when she was a doctoral student in my lab. And she chose to focus on the embryonic period of mouse retina development. And this she selected this time window because during this first wave of retina development, we're generating the early born retinal cell types that I mentioned. So retinal ganglion cells, cone photoreceptors, amacrine cells, horizontal cells, with retinal ganglion cells being the first cell type to be generated and abundant at this time. And it's a very simple system to ask, how do microglia interact with these different components of the developing nervous system? So at embryonic ages, only progenitors, these differentiating neurons, and microglia aren't present. At this stage, we don't yet have vascularization of the retina. The astrocytes have not yet invaded the retina. There are no oligodendrocytes, and we don't see evidence for other immune cells within the retina at these stages. And so the image on the bottom just shows an E14.5 mouse retina with the nuclei stained with DAPI here. And brain 3 is an immunostain for the retinal ganglion cells. And these are cells that are in the process of exiting the cell cycle and migrating down to form the retinal ganglion cell layer within the inner retina. And then the microglia here are labeled, in this case, with an immunostain for IBA1. And Sarah did an extensive quantitative analysis and showed that during retina development, these retinal microglia are preferentially associating with neurons and really intimately contacting these newborn retinal ganglion cells, even as they first exit the cell cycle and start migrating down to populate the retinal ganglion cell layer. And this was of interest to us because work from others in cortex had shown that the microglia actually seem to reside more in the progenitor layers and interact preferentially with progenitors. And so it suggested that microglia might be having a particular conversation with these newborn retinal neurons. Next. So to address the function of microglia in development, we chose to eliminate microglia and ask whether there were any effects on retina development. And this is a strategy that has been used by multiple other labs. And work from Kim Green's lab had shown that if you inhibit signaling through the CSF1 receptor, microglia are dependent upon that signaling pathway for survival. And so you can administer, for example, an inhibitor to CSF1 receptor, and that will deplete microglia. We chose not to take that inhibitory strategy because progenitors also express the CSF1 receptor. So we wanted an approach that would selectively target just the retinal microglia and spare the retinal progenitors. So Sarah obtained a floxed allele of the CSF1 receptor and then used a Cre-ER, an inducible Cre system, a tamoxifen-inducible Cre that's expressed selectively in the microglia and macrophage lineage, the myeloid lineage that was developed by Stefan Jung. And she used the ROSA TD tomato reporter in red there to mark which cells have active Cre. So in the top row here, you can see a wild type cross-section of a wild type retina that the microglia are labeled with IBA1 and are present. In the absence of tamoxifen, if we take a heterozygous animal, so we retain one normal copy of CSF1 receptor, the ROSA TD tomato will now mark the microglia and the vitreal macrophages, but they're not depleted since we still have CSF1 receptor signaling. Now, if we take a homozygous floxed mouse and deliver tamoxifen, we can see selective depletion of the microglia within the retina, but maintain the vitreal macrophages because they're not dependent on CSF1 receptor signaling for survival. So this shows us that our strategy is working. We can deplete 90% of the microglia in the developing retina, and we've done this at multiple stages. Next slide. So then we asked, what effect does this have on the development of the retina? And we ran a whole panel of different markers for various retinal cell types. And I'm just going to highlight the effect that Sarah observed on retinal ganglion cells. So again, this is, we've done the analysis at multiple stages, embryonic day 14.5, 16.5, as well as at birth. And Sarah consistently observed when she stained for brain 3 to mark retinal ganglion cells in red here, and then counted a consistent region of the retina, she could see an increase in the density of retinal ganglion cells when you eliminated the microglia. So ablation of microglia results in increased density of these retinal ganglion cells within the retina. Advance. And since microglia may simply be recognizing sort of cells that are in the process of dying, we actually waited for 4 days until birth, and then again counted the presence of retinal ganglion cells, and Sarah saw that these cells were maintained. So the excess retinal ganglion cells persist, they're viable, and they're not in the process of dying. Next. And to independently confirm whether microglia are involved in regulating retinal ganglion cell number within the developing retina, we used a different strategy, which is to administer minocycline to the pregnant dam. And minocycline is a tetracycline derivative. It's a widely used inhibitor of microglial function. And we confirmed that, in fact, it seemed to reduce the expression of activation markers of the microglia. And when we did this, we dosed with minocycline and again examined retinas at E16.5. We see an increase in retinal ganglion cells. So using both a genetic approach as well as a pharmacological approach, we found evidence that microglia would normally limit retinal ganglion cell number during development. And I'm not going to go through all the data. We didn't observe a change in other retinal cell types. So we think that this effect is selectively on regulating RGC number during this embryonic period of retinal development. Next slide. So how does perturbation of microglia increase RGC? So how does either ablation or inhibition of microglia increase retinal ganglion cells? So one possibility is microglia might be constraining the differentiation of retinal ganglion cells, and when you eliminate them, more retinal ganglion cells are born. Or microglia may actually be playing a role in promoting retinal ganglion cell death, apoptosis. So we set out to address that more directly. Next. So are more neurons being born when microglia are depleted? That's the question. Is that why we're getting an increase in the density of retinal ganglion cells? And to address this, we used classic approach of birth dating. So we inhibited the microglia, actually when microglia are inhibited. So we inhibited the microglia, then we pulsed with BRDU at E13.5, waited 1 day, and then collected the embryos at E14.5. Advance. And so when this happens, the BRDU will be incorporated into proliferating progenitors, and as those progenitors exit the cell cycle, that mark will be incorporated into brain 3 positive retinal ganglion cells. So we count the number of BRDU positive, brain 3 positive retinal ganglion cells, and that's a measure of the cells that have actually exited the cell cycle. And when we did that analysis, we did not see that microglia had any significant effect on the genesis of retinal ganglion cells. And we didn't see effects on other early born cell types or any effects on progenitor proliferation. So it did not seem like microglia were selectively modulating the genesis of retinal ganglion cells. Next. So do microglia regulate the differentiation of retinal ganglion cells? No, because our BRDU birth dating analysis shows no change in RGC genesis. Next. I'm not going to go through all the data, but we also analyzed whether microglia promote apoptosis. And when we did that analysis by looking at cleaved caspase 3, we saw no change in apoptosis, which is actually minimal during this embryonic period, and no change in the contact of microglia with apoptotic cells. So we don't think that that is a mechanism. Next. So how do microglia limit retinal ganglion cell number? Next. So maybe you can click the movie, Mariana. So microglia are phagocytic at embryonic stages. We've actually done in situ and gene expression analysis, and we show expression of, there should be at the bottom left of the image a little arrow to play the video. I don't know if it will play. And if not, I'll just explain it. So this is just an Imaris reconstruction of a GFP labeled of the actual lower left corner of the black square. There might be a video thing, and if not, it may not be embedded on the thing. So this is an image showing a GFP labeled microglial cell. We've stained for, it's fine. Don't worry. It's not essential. So we've stained for brain 3 to label the retinal ganglion cell nuclei, and we've stained for cleaved caspase 3 to label apoptotic neurons, retinal ganglion cells. While we do see microglia, it's okay. We've lost the presentation. Don't worry about the video. It may not be configured properly. So anyhow, what we see is that the microglia are in the process. This is just an example of microglia engulfing a brain 3 positive nucleus of a retinal ganglion cell. And this nucleus is non-cleaved caspase 3 positive. So we see multiple examples thank you. Yeah, it has to maybe not be in presentation mode. We see multiple examples of microglia engulfing these non-apoptotic retinal ganglion cells. And this was striking to us because Guy Brown and Jonas Nehar had observed in other contexts that microglia can participate in a process that they term phagoptosis, which is death and elimination of viable cells through phagocytosis, so recognition of a cell that's viable. And as I showed you, we believe that these cells are viable because when we eliminate the microglia, the cells don't go on to die. They actually persist and remain for several days. Okay, we can go back into presentation mode. Perfect. So we hypothesized that microglia may be recognizing newborn retinal ganglion cells, and we actually, through our BRDU analysis, were able to narrow down this window of elimination to about 24 hours after birth. Within 24 hours of being born, the microglia seem to be recognized and eliminated. So how might this be happening? So this is from a review of Brown and Nair, highlighting that microglia interact with neurons through multiple signaling pathways. And so we wanted to systematically address which of these signaling pathways might be involved in this process. And so advance, please. We inhibited the microglia with minocycline. So we administered minocycline since we wanted to maintain the presence of the microglia, and then analyze for changes in gene expression. And we found that inhibition with minocycline did not alter the expression of many of these well-defined cell-cell recognition pathways, such as MFGE8 or vitronectin receptor. So, and since minocycline did inhibit this process of RGC elimination and it maintained higher levels of retinal ganglion cells, we ruled those out as candidates in our process. Next. However, in the center, you'll see that complement signaling has also been implicated in this cell recognition process. And I'm just showing a snapshot of the qRT-PCR data that we generated, we find that both multiple complement components, C3 and C1q, as well as the ITGAM, the CR3 component of the CR3 receptor, seem to be dynamically modulated with minocycline signaling. So this suggested maybe complement could be a candidate pathway for mediating this recognition process. Next. This was of interest because I'm sure you're all very familiar with the elegant work from Beth Stevens and Dory Schaefer, showing that the complement pathway plays an important role in phagocytic elimination of synapses by microglia at postnatal ages, for example, in the tectum. So this complement pathway clearly can be an important mediator of phagocytic function for the microglia. Next. So we sought to ask whether complement expression, complement components were expressed in the developing retina at the time these retinal ganglion cells were being eliminated. So in the top panel is a whole mount view, flat mount view of an embryonic day 13.5 retina. The GFP is marking the microglia. The red brain 3 is marking newborn retinal ganglion cells. They are in the sort of at the peak of genesis at around this time. And C1q is shown in white there. And you'll see that not all brain 3 positive retinal ganglion cells, but a subset of them are expressing, do have C1q and they're marked by that protein. And then when we do a cross section of the retina at embryonic day 16.5, you can see in this case, in the brain 3 label, there are differentiated brain 3 positive retinal ganglion cells in the emerging retinal ganglion cell layer in the inner retina. And there are also cells that are migrating down that have recently exited the cell cycle, differentiated, and are migrating down to join that layer. And we see that these brain 3 positive cells are, a subset of them are marked with C1q, and it seems to be preferentially at that interface, either the migrating cells or cells that have recently joined the emerging retinal ganglion cell layer. So not all brain 3 positive retinal ganglion cells are marked with complement. And that was exciting because it makes sense. We actually only saw 10 to 15% of the cells look like they were being eliminated. And if you look at the GFP label, you'll see that the microglia seem to be clustered in that region and potentially preferentially interacting with those labeled cell populations. Next. So to test whether this process might be involved in the elimination, whether complement signaling might be involved in the elimination process, we collaborated with Dory Schaefer at UMass, University of Massachusetts in Worcester, and she provided embryos that were deficient for complement receptor 3, which is selectively expressed by the microglia. So the microglia have the receptor and are recognizing the complement. And when we analyzed those retinas, we found that loss of complement receptor 3, again, increases the retinal ganglion cell density at P0, similar to loss or inhibition of microglia. The effect is not as pronounced as when we actually eliminate the microglia, and that's consistent with the work that Dory had done on synapse elimination, and that there's likely additional pathways that are involved in that process. But based on this analysis, we think it supports the idea that this complement signaling pathway might be involved in selective elimination of the retinal ganglion cells. So how do we put this in the context of development of the retina? So many of you are aware that there's a large wave of retinal ganglion cell loss that occurs beginning at around birth in that initial postnatal period. That peak of RGC death is an apoptotic cell death, and it requires Bax signaling. So it's sort of a classic apoptotic developmental cell death. But there had been evidence in multiple species based on axon counts that there is an earlier there was evidence for earlier RGC loss, but little evidence for apoptosis. So, and then proportion of or number of retinal ganglion cells that are eliminated can vary widely depending on species. But we think that potentially this phagocytosis elimination process might be responsible for this earlier non-apoptotic wave of RGC loss. Next. So putting this into a little bit larger context and starting to develop the theme of the rest of the talk, are there parallels in disease? Complement dysregulation is a key feature of multiple degenerative diseases. So C1q knockout can be neuroprotective in glaucoma, and I'm going to talk about Alejandra's work on C3 inhibition. But there's also direct evidence that microglia can phagocytose non-apoptotic rods during photoreceptor degeneration, work from Wai Wang's group. And then microglia have also been shown to eliminate stressed but viable neurons after brain ischemia stroke models. Next. So microglia seem to be important and can dynamically respond to changing environmental cues and change their transcriptome. And I will pause here, Mariana. Would you like me to shorten the talk to help us stay within the allotted time? You're muted. I just...
Yeah, sorry. No, not at all. I think you can conclude your presentation.
Yeah, I tried to keep it...
Okay. If some people want to make questions and have to leave, they can write in the chat and they can ask and they can see it later in the YouTube.
Respectful of people's time. I appreciate it. Yeah, thank you. Okay, we can go back to presentation. So microglia can dynamically respond to changing environmental cues. They dramatically change their morphology. They change their transcriptome. They change the types of factors that they secrete. And so extensive effort now is focused on really trying to understand how microglia might be changing their transcriptional responses, both during development as well as in injury and disease. And so we thought this might help give us some insights into potential function. So this is now work that's underway, but I'll give a snapshot of what we've done so far. So we asked, do microglia change transcriptionally during development of the retina? We collected retinas at embryonic day 16.5, at postnatal day 7, as well as adult P60. And we chose those time points because embryonically we're generating these early born cell types, and I just told you that microglia are involved in elimination of retinal ganglion cells. And then postnatally, there's a whole series of important developmental processes, including developmental cell death, vascularization of the retina, development of synaptic connectivity, and the generation of later born cell types such as rod photoreceptors and Mueller glia. Next. So Sarah isolated retinas, dissociated them, and then performed FACS sorting to isolate microglia and excluding CCR2 positive cells that might be other myeloid cells, non-microglial cells. And she was able to selectively isolate a highly enriched population of retinal microglia at these 3 time points, and then performed RNA-seq analysis. Next. And when we took that transcriptome data and performed clustering, we could see that microglia clearly undergo sequential changes in gene expression from E16.5 to P7 to postnatal day 60. And they gradually acquire the expression of adult microglia genes, and that these core microglia genes are expressed. So we're confident we've been able to isolate that population quite selectively. And at early stages, these microglia are expressing cell cycle genes and cytoskeletal remodeling genes consistent with the active role that they're taking in development. But advance, we noticed that at postnatal day 7, that there was a peak of expression, and I think if you advance, it will come up, of these lipid metabolism and phagocytosis genes. These are genes like ApoE, AXL, LIS2, a lysosomal gene. And these are genes that are not normally expressed in adult quiescent homeostatic microglia as they're undergoing their surveillance functions. You can see at P60, their expression level is fairly low, and they seem to be transiently increased in this postnatal day 7 period. So this was quite striking to us. Advance. And we were able to perform in situ analysis with single cell resolution, and we're able to show that that there's considerable diversity of these microglia in the early postnatal retina. And we've now just completed a single cell sequencing analysis that has given us deeper insight into that diversity. I'm not going to go through that data here. We're still in the process of analyzing that. Next. But what really struck us about this early postnatal microglia signature is that this gene signature really shared features with rare subpopulations of microglia that had been identified in aging and neurodegeneration. So this was work from Olive Butasti or Ito Amit, comparing how microglia respond in multiple forms of aging and neurodegeneration, such as Alzheimer's disease or ALS. And so this cartoon just shows that homeostatic microglia express a set of genes that help them maintain their normal surveillance function. And in response to these perturbations and stressors, the microglia can shift their transcriptome, and they downregulate many of these homeostatic genes, and they upregulate these genes that are involved in phagocytosis, lipid metabolism, and sort of recognition of stressed or dying neurons. And this just shows that there are multiple stages in this transition that we are still trying to understand, but that an important signaling pathway, the cell surface receptor TREM2, and components of that signaling pathway seem to be involved in regulating that transition. And that's important because this signaling pathway has also been implicated in Alzheimer's disease. So developmental microglia are looking like microglia in aging and neurodegeneration. This was a surprise to us. So we wanted to understand it more fully. Next. So we started to look more carefully at some of the markers that were expressed in our postnatal retinal microglia. And one of the hallmarks of microglia that was an important readout to us was that these cells seem to upregulate expression of CD11C. They express higher levels of CD11C. And by harvesting and analyzing those CD11C high microglia selectively, we were able to show that these microglia are less homeostatic, so they downregulate homeostatic genes like TMEM119, and they upregulate these disease-associated genes. So a majority of the postnatal retinal microglia, 60% at P7, seem to be CD11C high. Next. And this was again striking to us because these CD11C positive microglia have also been implicated in association with plaques and Alzheimer's disease. So it suggests that there might be shared functions in both the gene signature as well as these cell surface protein markers between microglia in the early postnatal retina, as well as the changes that are observed during aging and degeneration. Next. So we wanted to understand whether similar signals might be important for driving this shift in gene signature. And so we obtained knockout mice for TREM2, as well as ApoE, since those pathways had been implicated in regulating the transition to disease-associated microglia in aging and degeneration. And we saw a partial effect on the proportion of these CD11C high cells that are present. And we saw that homeostatic gene expression was increased. So it looked like this might partially contribute to this regulating this transition, but clearly the effect was not very complete and really only accounted for a subset of this effect. Next. So we said, well, what are the normal cues that these microglia might be responding to? Are there signals in that environment that might be driving these cells to adopt this specific gene signature? And apoptotic cell death. Can you go back one, please? Apoptotic cell death is prominent in the developing retina and during that neurodegenerative disease. So we asked, is this a cue for microglia? So I think you can advance now. So to ask this, we obtained Bax knockout mice. And so Bax is a key component of the apoptotic signaling pathway. And in a Bax knockout mouse, the neurons that would normally degenerate developmentally are maintained. So there's no developmental cell death of neurons. And Bax is redundant with another family member in microglia. So we didn't think that there was going to be a direct effect on microglia by looking at the Bax knockout. And when we looked at the Bax knockout, we saw that expression of these disease-related genes was reduced. And so in this in situ here, you can see that Liz2 and ApoE expression is reduced. And then also by qPCR, we saw reduced expression of multiple markers of this disease signature, as well as phagocytosis genes such as CD68, and increased expression of homeostatic genes like P2RY12 and TMEM119. So we think that in the absence of that normal developmental cell death, that the microglia are lacking the cue that would normally drive them to become this more phagocytic remodeling phenotype, and instead remain in this more homeostatic surveillance mode. So this highlighted that this might be an important pathway. And then we performed flow cytometry and also saw that the proportion of these CD11C high cells was also reduced in the Bax knockout. So this reinforced that we were sort of shifting the phenotype of these cells in a substantial way. So retinal microglia, we conclude that retinal microglia have distinct transcriptional states across development, and we're now getting single cell resolution with that. And that in this early postnatal period, there's a significant proportion of microglia that resemble microglia in aging and disease. And this has actually also now been reported by Beth Stevens as well as Ben Barris's lab in brain. And those microglia in that case seem to be associated with developing white matter tracts. Here we find that developmental cell death is a major driver of the disease-related signature in this postnatal retina, since we see that it's reduced in a Bax knockout animal. Next. And so in the last part, I will go through this quickly, since I know we're running late, but I wanted to close the circle and say, you know, we're very interested in how microglia are functioning both in development but also in disease. And we think that our work in the developing retina may actually begin to help us gain some insight into some of the pathways and processes that might be involved. But we also want to directly study disease. And an important disease of the retina is glaucoma. It's a progressive chronic neurodegenerative disease. These retinal ganglion cells, which are the major projection neurons to the brain, progressively decline and die, and the optic nerve itself degenerates. And it's the second leading cause of blindness in the US. And age is a major risk factor, as is elevation of intraocular pressure, which can put stress on the retinal ganglion cells and components of the optic nerve head. Next. So this is a summary from Adriana DiPolo showing that this RGC decline is a progressive process. So retinal ganglion cells project from retina to brain under the and in healthy conditions, they're myelinated axons within the optic nerve. As in the early stages of glaucoma, there's evidence for progressive decline of these neurons before they actually die and are eliminated. So there is impaired axonal transport, there's gene silencing, there's retraction of dendrites, there's synaptic pruning and elimination and sort of loss of connectivity. And then the end result of that is somal apoptosis, axon loss, and elimination of the retinal ganglion cells. And so we're very interested in the sort of the trajectory of this decline, and in particular how microglia and the signaling pathways that they respond to might be involved in this process. Next. So this is just showing an example. This is from Alejandra Bosco's work. She's here prepared a flat mount retina, adult retina, and this is using the DBA2J model, which develops age-dependent progressive degeneration. I'm going to introduce that in a moment. And it shows that this process is asynchronous. So you can have regions of the retina that are healthy. Here the axons are labeled in red for phosphoneurofilament, and the soma of the RGCs is in green. You can have a healthy region, and directly next to that, you can have a degenerating region where there's loss and degeneration of axons, fragmentation, as well as loss of the brain 3 positive retinal ganglion cells. So it's a asynchronous progressive process. And then within the nerve itself, we have loss of these beautiful myelinated axons and axon bundles that results in the loss of connectivity to the brain. Next. So the model that Alejandra was working with is the DBA2J model. This is a inbred mouse strain that was developed and well characterized by Simon John's lab at Jackson Labs. And this, in this model, the disease shows variable onset, and it shows progressive sectorial degeneration. This means that different sectors of the retina are progressively impacted at different times, which is similar to what is observed in human disease. And there's a strain-matched control substrain that has a normal copy of GPNMB, which is a component of the pathogenesis in this model, and it doesn't develop glaucoma. So that's the control strain that we use. Next. So in the DBA model of glaucoma, as I said, it was age dependent. So at about 7 months, the majority of retinas and optic nerves are healthy, although we do start to see some evidence in some retinas for early stages of decline. And then over the period of the next months, we start to see increasing evidence for both retinal ganglion cell damage and loss, as well as optic nerve damage and loss that's progressive. So by 12 months, the majority of the optic nerves and retinas are in this degenerative state, and that proportion increases over time. And by 15 months, it's quite severe in most eyes. Next. So Alejandra first asked early on, how do microglia respond through this course of degeneration? And she was able to show that microglia within the DBA2J retina will cluster very early at this central optic nerve head region at ages that precede frank RGC degeneration. So microglia are very sensitive sort of biosensors. And this central optic nerve head region, there's evidence in both humans and mouse that this is an important site of early stress and axon damage. So this clustering was evident in the DBA2J model, but not present in the control's GPNMB substrain. Next. And then using the GFP reporter mouse, fractalkine GFP reporter mouse, Ali was able to also perform live on confocal scanning laser ophthalmoscope analysis in a living mouse to scan the retina and detect the GFP expressing microglia. And she did this at multiple sequential time points as the animals were progressing towards this glaucoma pathology. Next. And so you can see in the central, the lower panel here, the central region, you see this clustering and hypertrophy of the microglia, which is evidence of increasing activation. And we also know that there's infiltration of macrophage and other myeloid cells within this core region. That's work that Gareth Howell's lab has contributed. Next. So she was able to monitor this and also confirm it by ex vivo confocal analysis. And her analysis indicated that this early clustering of GFP-labeled cells was a harbinger of later pathology and very tightly correlated with those retinas that were then going to go on and progress to develop disease at a later time point. Next. So what are the molecular pathways potentially involved? So I've already mentioned the complement system. So this is now showing you, it's a well-defined innate immune pathway. The complement system is a potent system for activation of microglia. And it just shows that there are multiple branches, both the classical alternative as well as the lectin pathway, for leading to activation of C3 convertases and both deposition of C3 fragments, such as C3d, as well as activation of the membrane attack complex. In DBA2J, this C5 convertase step is absent, so we are entirely focused on this, the C3 convertase step and the signaling that results through that activation step. Next. And we had seen early on in our first microarray analysis of the DBA retina in 2006 that complement is C1q here is significantly upregulated in the DBA2J as these retinas progress from early to later stages of disease. And then Gareth Howell and Simon John's lab has done an extensive microarray analysis and also found multiple complement components implicated. Next. So there's now evidence, and I'm on the right side here, from multiple labs, both in human, DBA mouse model, as well as acute ocular hypertension models, that there is significant dysregulation of complement signaling in glaucoma, and that these early components, C1q and C3, upregulate early in the retina, and that this is then coupled to functional decline and neurodegeneration. And Simon John's lab has generated global knockouts of C1q as well as C3, and C1q is protective, whereas the C3 knockout seemed to have somewhat of an exacerbation on the process of degeneration. So we don't yet have a full understanding of exactly how a complement might be impacting the neurodegeneration process. Next. So Alejandra specifically performed immunostaining for the C3d fragment, which is, and found that this is deposited on the inner retina in this DBA2J glaucoma model. And this was localized to retinal ganglion cells, microglia, as well as astrocytes. And this was not observed in the control stain. And C3d is significant because it's an important recognition molecule for microglia, and it may be involved in some of the remodeling events. Next. So she asked, how can we potentially restore this complement balance in the retina and test the impact on glaucoma progression? And so we collaborated with Steve Tomlinson's lab at Medical University of South Carolina, and they had developed a very clever approach to selectively express a C3 inhibitor, SCRRY. This is a naturally occurring inhibitor in rodents. And to increase the efficiency of this inhibition, he coupled it to the CR2 recognition receptor. And so that CR2 recognition receptor will essentially allow this inhibitor to be selectively targeted to the sites of complement deposition. And then the SCRY will inhibit C3 convertase formation, inhibit C3 cleavage and activation. So you should reduce the C3d deposition. So this CR2 will guide this inhibitor to the sites of activated C3 deposition, and it's been shown to increase the efficiency of the inhibitor in other models of damage and disease by 10-fold. Next. So we obtained this construct, and we collaborated with Bill Hauswirth's lab at the University of Florida to have this packaged in AAV2. We performed intravitreal injection at 7 months to deliver it to the inner retina. Then we waited for several months and then analyzed at 10 to 15 months for effects on glaucoma pathology. And we were able to confirm stable expression of the fusion protein. I'm not going to show that. Next. And I highlight this because we benefited from the really important work of Hilda Peters Silva when she was with Bill Hauswirth. We used the high efficiency version of AAV, the 4YF mutant capsid version, and this resulted in very stable expression and transduction. Next. As others have shown, when you perform intravitreal injection, this is just AAV GFP control, we get very efficient transduction of retinal ganglion cells and very persistent long-term transgene expression. We wanted just to confirm for ourselves that this was working effectively for us, and we can even see the GFP extending out into the optic nerve. So we're convinced that the retinal ganglion cells themselves are transduced. Next. And so when we delivered the AAV2 CR2 CRY inhibitor through intravitreal injection, I'm going to just kind of walk you through what we see. So in a normal 7-month retina, in a naive uninjected DBA2J, we normally don't yet see much C3d deposition, and SMI32 labeling shows relatively intact retinal ganglion cell axons. Next. By 10 months, in many retinas, it's asynchronous, so it's not always at the consistent time point, but by 10 months, we see evidence for this C3d deposition, as well as a loss of these neurofilament SMI32 labeled axons in many regions of the retina. Next. And then when we deliver this inhibitor, we consistently see reduction in C3d deposition at this 10-month time point and preservation of these neurofilament labeled axons. Next. And so when we actually quantify whether the effects on preservation of the retinal ganglion cells and optic nerve, first we looked at the optic nerve degeneration. And we can score the nerves based on whether they are healthy, declining, or degenerative. So healthy in green, declining in yellow, or degenerative in red, where more than 50% of the axons are gone. And we can see normally from 7 to 10 months that there's an increase in the proportion of degenerating optic nerves. And at 10 months, when we now deliver the inhibitor, we see a much higher proportion of healthy optic nerves and fewer degenerating optic nerves. Next. And then if we wait till 12 months, because the disease does progress, now we see an even higher proportion of degenerating optic nerves and untreated eyes, and that this is reversed, and that progression is halted in the treated eyes, or slowed. I would say that the progression is slowed. And then next. And then even if we wait 3 more months, we still seem to maintain a significant proportion of healthy optic nerves. And this is at ages when the vast majority would normally be highly degenerative. Next. And if we also look directly at the retinal ganglion cells and score for loss of brain 3 positive retinal ganglion cells, or expression of that marker, we can see at 12 months that there's a significant preservation of portions of the retina that have no depletion of retinal ganglion cells after treatment. Next. So our conclusion is that this C3 targeted retinal gene therapy seems to delay the onset and slows and then ultimately halts the progression of neurodegeneration. Normally, we would have progression from 10 to 12 to 15 months, where a large proportion of those retinas would be impacted and optic nerves. And when we treat with this complement inhibitor, we seem to delay the onset and slow and then ultimately halt the progression of this degeneration. So we are now in the process of closing the loop and trying to analyze how complement inhibition might alter the microglia responses in the retina. We have some evidence that there's significant transcriptional changes, and we are also analyzing this in a distinct model of glaucoma that Alejandra is working on and developing. Next. So the theme that I just wanted to highlight is that microglia are known as professional remodelers of the central nervous system. And we have been interested in understanding their function in both development as well as in aging and disease. And we think that we can learn something by analyzing their function and signaling pathways in both contexts. So in development, we see evidence for high expression of disease-associated genes and lower expression of homeostatic genes, and that as they transition to become adult homeostatic microglia, this sequence of gene expression flips, and then it gets redeployed again as microglia respond to challenges due to injury or damage or degeneration. And so we hope that by going back and forth, we can start to dig into some of the triggers and signaling pathways that might be involved in that process. Next. So this is our campus. It doesn't look like that right now since we're in summer, but I like this picture because Jason Shepard is a young faculty member in my department, and he's also an avid photographer. So he managed to capture this picture from the hillside looking out over campus. It's been a really wonderful and collegial place to work. Next. And all of this was, of course, made possible by a remarkable team. And so Alejandra, I will highlight first, many of you will recognize her. She's a real friend and wonderful colleague. And she has made just tremendous contributions to the work in the lab and is a real leader within the group and is really developing exciting directions, really digging into pathways involved in glaucoma progression. Sarah Anderson, next to her, is she was a PhD student in the lab, and she recently graduated and is now staying on for a short period of time and is really contributed to the analysis of RGC elimination and transcriptional regulation, and is now looking at single cell analysis and specific pathways that are involved in that elimination process. And then the other people I want to mention are Cesar Romero, really worked very closely with Alejandra, did a lot of the imaging and mouse work for the glaucoma project. Jackie Roberts did a lot of the bioinformatics and gene expression analysis work. And we just always benefit from the amazing team of people that we have here. And then Dory Schaefer is the collaborator from University of Massachusetts who helped provide the CR3 knockout animals. And I just wanted to thank you for your attention very much and apologize for the delayed start. And I'm happy to take questions.
Thank you very much, Monica. I really enjoyed your presentation. I was out.
Peraí, tô com negócio ligado.
Yes.
Puxa vida.
And I'll take a look here if we have any questions. But if anyone that is at the Google Meet want to make a question openly, you can just turn on your microphones and address your question.
I have question.
Okay.
I have.
Júlio Sacrosanti, do you? Feel free to do
Okay.
it.
Dr. Vettet, thank you very much. I learned a lot. I'm not a neurobiologist, so I may have questions that are a little bit, view from my perspective as an immunologist. I think that a major contrasting picture that you have described between the developmental stage and glaucoma or aging process, which allows for pathological processes, is the absence of vascularization during development, which you very nicely stressed in your presentation. So my question is, for the physiological role of the microglia, phagocytic activity involving opsonization of the retinal ganglion cells, assuming that the ligands are C1q, perhaps even C3b, I wonder how is the regulation, the endogenous regulation of complement activation takes place during development vis-a-vis what happened during the pathological circumstances that you have described so nicely in the model of glaucoma. Because in that situation, I would assume that probably you have at least a low level of leakage, which could precipitate a very, very important activation of the cascade. So the demand on the endogenous regulation should be much higher. So it's not clear to me whether the susceptibility to the glaucoma in this model has to do with differences in the vascularization, hyper susceptibility to leakage, or maybe it has to do with the fact that you may have a low-level expression of DAF or many other regulators of the complement cascade, including soluble components such as C4 binding protein, for example, which could play a role in that critical stage. So it's, from my perspective, it's very intriguing, everything that you have described. I wasn't aware about that, so it was a beautiful lesson.
Thank you. So I think you've raised a really important question that we're very interested in. And one of the attractions of thinking about the developing retina is that there is so much complexity in the adult nervous system with the vascular components, changes in permeability, infiltration of other cell populations. This was, this was a chance for us to maybe hone in a little bit more specifically on this more direct interaction between, early born neurons and the microglia. There's still a lot that we need to learn. One of the questions is, what is the source of the C1q? Because we know from studies in multiple groups that C1q is highly expressed by microglia themselves. And when we perform immunostaining, we definitely see that microglia are expressing C1q. And so are they simply coming along and tagging those retinal ganglion cells and sort of choosing which ones need to be eliminated, because clearly not all of them are. The other possibility is that that there might be regulation of C1q by the retinal ganglion cells themselves. And there is evidence from Beth Stevens' group that TGF-beta signaling in retinal ganglion cells can stimulate expression of C1q. And we actually, in sort of unpublished work, when we deplete or eliminate the microglia, we can still see evidence that C1q is marking the retinal ganglion cells. So it would argue that there may be a component of that expression that's coming from the neurons themselves. And so then how are they actually being selected and targeted, and why are only a subset involved? One idea that we're interested in is whether this might developmentally might be an example of cell competition or sort of survival of the fittest, and a subset of the cells may be less robust, metabolically challenged in some way, and they tag themselves for elimination. We want to look at some additional components. This is sort of there's evidence for these being very active processes in fly development, for example. But maybe this is an opportunity for us to look in a very simple defined system at how you select and remove cells developmentally. We do think it's part of the normal process, and it seems to potentially be happening to different degrees in different species. Potentially it's an opportunity to kind of work on pathways and mechanism.
Thank you, Julio. I think Rafael Linden has some questions as well. Rafael, if you want to.
Yes, Monica, thank you very much for, can you hear me? Thank you very much for the talk. It was really exciting, very interesting work. The whole work in the lab. I have been following only some of it, but it's really impressive. I have three short questions, perhaps. The first one would be whether you have looked at the role of macrophages at the surface of the developing retina, the cells that look like normal blood-derived macrophages, and they are involved in phagocytosis and apoptotic cell bodies. Is there anything you can say about that in view of the impressive, very early role of microglia culling the retinal ganglion cell population?
I think that's a really important point and something we're really trying to think hard about. Early on, there weren't terrific markers that distinguished microglia from macrophages. So, for example, IBA1 will label both cell populations. Now our sequencing analysis and the single cell sequencing analysis, both in the retina as well as the work from others in brain, are revealing much more robust markers that will help us really mark and distinguish those cell populations. The one thing I can say during that embryonic period is that the depletion strategy selectively eliminated the resident microglia, but not the macrophages because of their differential dependence on CSF1 receptor signaling. So, we were able to confirm that those vitreal macrophages were still present. So we're confident that this embryonic phagocytic elimination is mediated by the resident microglia. And we don't see evidence that those vitreal macrophages are then invading the retina under those conditions. But in the postnatal retina, we think the situation is much more complex. And we are seeing evidence for a proportion of cells that express these macrophage markers, and we just now have some students starting to do that staining analysis and figure out where they're distributed and which cell populations or structures they might be interacting with, especially since that's when we're really starting to develop the retinal vasculature. So I think that's going to be really exciting to kind of think about the interplay between these two different populations. So we're hoping to understand that better, but I think it's extremely important.
Thank you. I have two more questions, but I don't want to interfere with other people. Mariana, what do you think?
You have the right to ask one more, and then we go to the YouTube, to the questions from YouTube, and then we can come back later if you want to ask.
Okay.
Who's your favorite?
I was wondering, what do you think? I recall, and I never actually heard much about that later on, but I recall several years ago, Lucia Valley-Resta suggested that the loss of retinal ganglion cells starting from precursors was on the order of 90%. So 9 in every 10 retinal ganglion cell precursors would eventually die. I wonder, how do you stand before this idea, whether it's dead, because I actually haven't followed the story, because the effect of the microglia, although that's of course in a single experimental setting, is relatively small. When you get rid of the microglia, you increase by a small proportion of the number of the ganglion cells.
Right.
It's very robust, but it's still a small proportion. I wonder whatever happened to all those missing cells, if they exist.
So the remaining kind of loss of microglia is driven in large part by this massive wave of RGC cell death that's happening after birth. So that's sort of why I highlighted that cartoon. There's this smaller non-apoptotic loss during this embryonic period. And then there's this massive loss in that early postnatal period, and a huge proportion of the retinal ganglion cells that are generated are eliminated at that time. And we think microglia are probably involved in recognizing and eliminating those cells that are apoptotic at that stage. We just highlighted the embryonic stage, and we know that represents a relatively small proportion, but it's intriguing that maybe in other species that proportion of cells may be different. Like I think it was in cat where there was evidence for much higher proportion of non-apoptotic cell loss. And so it was an opportunity for us to identify a biological mechanism for elimination of neurons that didn't necessarily involve apoptosis. And the ultimate complement of cell types in the retina may involve regulation of lots of different things: proliferation, elimination, cell death. And so it's one piece in this larger puzzle of how you shape the tissue. You know, there is evidence in the olfactory bulb that selective elimination of newborn neurons through a microglia-mediated elimination process is involved in odor sensitivity, so that there's sort of changes in the differential odor sensitivity. So in different parts of the nervous system, this may actually help shape function. We don't have insight into that in this case. So,
right, thank you. Thank you very much, and thank you for the talk.
Thank you.
Thank you, Rafael. Hilda is looking at the questions from the YouTube. Hilda, could you?
Yes, there's a couple of questions here. The first one from Claudio Serfati. Could microglia be implicated in the generation of a regular distribution of ganglion cell types as described earlier by Vessel?
So I want to make sure I'm understanding the question correctly. We're generating a diversity of different subclasses of retinal ganglion cells.
I guess so.
Is that, yeah. And I think that's actually a really interesting question. We have not dug deeper to look at whether there's sort of selective targeting of different types of retinal ganglion cells. There are now tools available to make it possible to distinguish some of these subclasses, and the single cell analysis that's been published by Josh Sain's lab and others has now given us an opportunity to maybe ask that question at a level of resolution that wasn't possible before. So I think that's would be potentially interesting and worth looking at, whether it's agnostic to subtype or not. So
there's another one. Can I continue? Sure. The next one is from Pedro França. The question is, did you perform any visual or ERG experiments on those AAV animals with reduced degeneration? Any functional analysis, I guess he's asking.
That is a terrific question, and it's something that we've been focused on for some time. It's quite challenging to perform the visual analysis in the DBA model. It's not an ideal system for visually guided behaviors. Those mice have other challenges, including corneal opacities that make it difficult to do visual function analysis. Dr. Porciotti's group in Miami has done pattern ERG, but we didn't have access to that level of analysis. And so what we are doing now is developing a different glaucoma model that relies on manipulating pathways that have been implicated in human disease. Alejandra is getting really nice evidence for intraocular pressure elevation and degeneration, and we're now going to use those mice to test this pathway and also perform more extensive functional analysis. And that will then not be hampered by some of the limitations we have working in the DBA model.
Hilda.
Posso? Tem mais. There's one more.
Yeah, sure. Go.
É do João Menezes. What happens postnatally with the first wave of RGC that are not eliminated embryonically when microglia is inhibited or eliminated?
So that's a really good question, and we actually are just in the process of doing that analysis. And so we have both done a global elimination of the microglia, as well as we've selectively targeted just the CD11C high population using, there is a CD11C diphtheria toxin mouse, and so we can selectively ablate that subpopulation with the idea that that subpopulation may be particularly phagocytic or involved in the remodeling processes. And so Nate Gaina in the lab is now, with Sarah, is in the process of doing that analysis and really looking systematically at multiple aspects of postnatal development, apoptotic cell recognition, vascular remodeling, sort of synapse remodeling. So those we think are, I think it's going to be complex, and there may be multiple roles that those cells are playing. So we're hoping to really tease that apart. So thank you.
Okay, I will get.
I have
just
one last.
Yeah,
the one you want.
Just one moment. We have two other questions from the chat here. I just one is a very quick one from Bruce Lima. Do you think that microglial phagocytosis of RGCs during development might be a maladaptive process?
So, I think that's always a judgment call. It's sort of
it's so tricky.
It's an interpretation. Clearly it happens during normal development, and I think that normal development is optimized to try to achieve a certain outcome. You're trying to balance the complement of different cell types, and so I would have a hard time thinking about it as a maladaptive process. It's just one of several developmental mechanisms for achieving sort of the normal number and complement of cell types. And I don't think it hurts the retina still seems to be reasonably functional when in the complete absence of microglia. So Wai Wong has done that analysis, and there's still visual function. So I think this is a refinement process that helps really kind of shape. It's not an all or nothing thing. I think it's really just kind of helping to shape adult function.
Perfect. And Viviane made two questions. I will choose one of them to ask. She was talking about what would be, in the second part of your presentation, what you believe would be the signal related to the migration of microglia to the optic nerve head. So to have an idea, like a chemotatic signal that would attract this microglia to the optic nerve head before RGCs degenerate.
That is a really important question. So I think that, we, and I think other labs are also very interested on, what those signals are. Are they coming from astrocytes in the optic nerve head? Are they coming from retinal ganglion cells? Are you recognizing damage signals on axons? Are there vascular components involved? So I think that is a really interesting and important question. And we're also very interested in what are the chemotactic signals that are helping drive microglia into the retina in the first place, both the retina and the nerve, and how do you sort of establish that normal population, because these cells are migrating in, and that's not fully understood, and certainly not in retina. So that's an excellent question, and I don't have the answer. So there is some evidence that various chemokines can be chemoattractive for microglia, that they will respond robustly to ATP release, for example. When cells become damaged, they can release ATP, and the P2RY12 receptor is an important recognition of these purines. So I think there could be multiple signals that are involved, including cell surface markers like I highlighted in the talk.
Yeah, just before letting Rafael make his last question, you just mentioned about the astrocytes, and this week the group of Chen Lidelo highlighted the relevance of this microglia astrocyte communication in RGC degeneration in some glaucoma models that they used. So what do you believe is the role of astrocyte in this? You just mentioned one possibility, but he mentioned that the astrocyte participation would be key to the degeneration, but there are other groups that say the opposite. So what's your opinion about this?
I think, the strength of Sean Lidelo's analysis is that they've really highlighted these different subpopulations of astrocytes, these A1 versus A2 astrocytes, and really show that that astrocytes, just like microglia, are highly responsive to environmental conditions, and that there are really important cues that will, including C1q, that will help shift the phenotype of astrocytes, and that will change the crosstalk between astrocytes and microglia and the neuronal components. And so in a complex disease like glaucoma, we do need to think about not just one or two cell types in isolation. We do really need to think about the network of cell types involved. So I think his analysis is really compelling and really hones in on a very specific response of astrocytes and the subpopulation and how they contribute. One of the reasons we tried to focus in the developmental model is we could kind of reduce down the system to a simplicity that might allow us to ask more direct questions about interactions between a very limited set of cell populations, recognizing that in the adult, you add in many layers of complexity, but in order to really dissect molecular pathways, that system had some advantages. So, yes, there's many players involved in this, and I think that makes it exciting, but also very challenging to solve. So,
I agree. Thank you, Monica. Rafael, you have your last question.
Okay. I was wondering, in view of the results that you have and the changes in the properties of the microglia, how do you see your results in view of that idea that you have an M1 and an M2 population of microglia cells, just like in the immune system in general?
So I think I would say in the last 3 to 5 years, the field has really dramatically moved away from thinking about M1 versus M2 as a classification system for microglia. And that was sort of largely based on analysis that had been done on macrophages and then trying to look for parallels in microglia responses. There have been, like Marco Prince and Keteman and others have written reviews sort of arguing that microglia, we know that from single cell analysis that there's a quite considerable complexity in microglia phenotype. So you can have up to 12 to 14 distinct clusters. Like Beth Stevens had done a single cell analysis in Developing Brain and sees, over a dozen distinct clusters of microglia with different gene expression profiles. And so I don't think it's an either or. Microglia are highly local responders to their microenvironment. And so this level of resolution of this single cell analysis is giving us insight into the considerable diversity and complexity in these responses, and also how plastic these cells are. You can nudge them in different ways, and you can dramatically shift their responses. And so some of these are clearly protective, and others can have sort of destructive consequences. But even the process of phagocytic elimination of material is likely a beneficial and important and protective process. So there's evidence from, human mutations in Alzheimer's risk genes that impact those important recognition and phagocytosis pathways, that if you disrupt that recognition and phagocytosis, you have excess amyloid accumulation and increased neurodegeneration. So I think we have to force ourselves to think more deeply about the complexity of how these cells are responding and what they're doing, and it's highly context dependent. So that's interesting and challenging for us as biologists, I think, to grapple with that.
Thank you.
Okay, so thank you very much, Monica. I know I'm sure everybody, including myself, learned a lot from your talk, and it was a pleasure to hear you, and it was very good to have everybody here. Thank you, all the students and professors that could participate and make these intriguing questions, and I hope you've enjoyed it as well.
I sure did. Thank you. And congratulations again. I'm so glad you're taking the time to celebrate this important time for your institute, and I wish you much success moving forward. Thank you all for this opportunity.
And next week, we'll have the chance to hear João Helvas from the University of Porto, the I3S Institute of Research. And he will probably talk some aspects of it, some subjects that we already discussed today, and microglia is one of his interests. So maybe you could join us next week again to listen to what he has to do. So thank you very much. And Pedro, I don't know if you want to say something, or I don't know if Bruno is still here.
No, just thank you again, Dr. Monica, for your brilliant talk, and for the students, just switch to Português para quem ainda está na sala e lembrando que na semana que vem, eu acabei não tendo tempo de avisar hoje, mas lembrando que só conta para crédito os alunos que estiverem participando aqui pelo Meet, não conta pelo YouTube, tá bom? Mas aí já vale para a semana que vem. So thank you again, Dr. Monica. Okay. Thank you, Mariana.
Thank you
very much.
Your patience with my technical limitations. I apologize for that.
Don't worry, this happens.
Bye-bye.
See you soon.